Today we will present the coupling and repulsion configurations of the dominant or wild-type alleles in a particular species of butterflies. In the coupling configuration, the wild-type or dominant alleles are found on one chromosome and the recessive or mutant alleles on the other. This is also referred to as the cis orientation. On the other hand, in the repulsion configuration, the wild-type alleles and the mutant alleles are found on the same chromosome. This is also known as a trans orientation. In the particular butterfly species, consider two alleles, big B and big L, which code for the wild-type traits, blue-colored wings and line pattern wings respectively. Blue is dominant with respect to white, and lines are dominant with respect to no lines. When a heterozygous individual with blue and lined wings, big B, small b, big L, small l, is test-crossed with a homozygous recessive individual, small b, small l, the progeny are as follows. Our question is, are the genes linked? What are the expected versus observed frequencies? The next question is, are the genes in coupling or repulsion configuration?
For the first part of the question, we can see that since the parental capital B, capital L, small l is heterozygous for both traits and other parent is homozygous recessive, in the absence of linkage, we expect a 1-to-1 ratio for each trait independently. This will lead to a 1-to-1 ratio for all possible combinations of the progeny. Looking at the actual offspring numbers, we see significant deviations from the expected 1-to-1 ratio. We can see that the higher numbers are for the progenies with blue color wings and no line patterns on them, and the progenies with white color wings and line patterns on them. This suggests that the genes for wing color and pattern might be linked. So at the end, we can say that yes, these genes are linked, as both the expected and observed ratios are not the same.
We know that one parent is heterozygous. However, we do not know whether the dominant alleles are in cis or trans conformation. Cis would look like this, and trans would look like this. Here we can see the heterozygous parent produces 2 non-recombinant gametes and 2 recombinant gametes, while the homozygous recessive parent will only produce 1 gamete. 2 of the possible 4 phenotypes of the progeny are recombinant, and 2 of them are non-recombinant. To determine if the heterozygous parent's alleles are in cis or trans conformation, we need to identify if phenotypes of the non-recombinant are the same as either of the parents' phenotypes or if they are different. The phenotypes of the parents are blue with lined wings or white with non-lined wings. The phenotypes of the non-recombinant progeny are blue with non-lined wings or white with lined wings. We can see that the phenotypes of the non-recombinants are different from the parents. This means that the alleles are in trans conformation on the heterozygous parent's chromosome. Therefore, this is an example of repulsion, meaning the wild-type alleles are not on the same chromosome, and it can be drawn like this. Since they are in repulsion, the genotype can be written like this.
This is the end of our presentation. Thanks for watching.